Plants are amazing living organisms with a unique ability. They can regenerate. This means that a single plant cell, under the right conditions, can grow into a whole new plant. Tissue culture takes advantage of this remarkable ability. Tissue culture is a technique where small pieces of a plant, called explants, are grown in a controlled environment outside of the plant. These explants can be anything from cells to tissues to organs. They are placed in a sterile nutrient medium that contains everything the plant needs to grow. The nutrient medium is a kind of plant food that is specially formulated to support plant growth. It contains essential nutrients, vitamins, and hormones. The controlled environment ensures the right temperature, light, and humidity for the explants to thrive. One of the most common applications of tissue culture is micropropagation. This technique allows us to produce a large number of identical plants or clones from a single mother plant. This is particularly useful for plants that are difficult or slow to propagate through traditional methods. The process starts with selecting a healthy mother plant. A small piece of plant tissue, like a leaf or stem segment, is carefully removed and sterilized to prevent contamination. This explant is then placed in the nutrient medium where it starts to grow. Under the right conditions, the explant will develop into a mass of undifferentiated cells called callus. Callus has the potential to develop into any plant part, like roots, stems, or leaves. By manipulating the hormones in the medium, we can direct the callus to form new plantlets. The ability to grow plants in a controlled environment offers numerous benefits for agriculture, horticulture, and conservation. One of the major advantages is the rapid production of disease-free plants. This is particularly important for crops that are susceptible to viruses or other pathogens. Tissue culture also allows us to conserve rare and endangered plant species. By preserving plant tissues in a lab, we can safeguard the genetic diversity of these plants and prevent their extinction. This is crucial for maintaining biodiversity and protecting our planet's natural heritage. Moreover, tissue culture enables us to grow plants outside their natural environment. This has opened up new possibilities for cultivating plants in areas with limited arable land or harsh climatic conditions. While tissue culture is a powerful technique, it does come with its challenges. One of the main challenges is contamination. Since the explants are grown in a sterile environment, any introduction of bacteria or fungi can be detrimental to their growth. Another challenge is the cost associated with setting up and maintaining a tissue culture laboratory. Specialized equipment, trained personnel, and high-quality reagents are essential for successful tissue culture. Despite these challenges, tissue culture holds immense promise for the future of plant science. Researchers are constantly working on developing new techniques and applications for this versatile tool. From developing disease-resistant crops to producing novel plant-based products, the possibilities of tissue culture are vast and exciting.